Discovery. I'm your gerbil host, not to be confused with the mouse, which is today's topic. Did you know that the human body is home to more prokaryotic bacterial cells than cells that make up the structures of your body? You have about 100 trillion microbial cells in and on you. Bacteria were the first organisms to appear on the earth, and while many of the bacteria found in your body are essential to human survival, incorrect proportions of certain types of bacteria can be hazardous to the body's overall function and health. Here I have two world-renowned scientists to discuss their research on bacteria found in the guts of mice.
We were faced with the question of how gut microbiota changes over time and evaluated the idea of composition in the gut bacteria can actually accelerate the process of aging by a process called inflammaging. We sought to uncover that of strategies that alter the gut microbiota composition in the elderly, if they could also reduce inflammaging and promote healthy aging.
We use both older and younger mice as our subjects. Our studies suggest that chronic low-level inflammation in these older mice is due to imbalances in gut bacteria. We see that inflammatory responses from gut bacteria are linked to age-related conditions such as stroke, dementia, and cardiovascular disease.
The elderly are known to have an altered gut microbiota composition. We orally transferred gut bacteria from old mice to younger germ-free mice and analyzed the changes in bacteria characteristics over a period of 4 weeks. We then compared these mice to a group of germ-free mice that were given bacteria of the young mice.
At the end of the 4-week period after bacterial transfer, we analyzed tissue from the immune system taken from the spleen, lymph tissue found in the small intestine called Peyer's patches, and lymph tissue found in the large intestine in mesenteric lymph nodes.
Now I'm sure you're all curious to see the data of the experiment and find out if they were able to deem their research significant. Let me direct you over to the graphs.
The microbiota in the old mice were shown to enhance the differentiation of CD4 plus T cells in the spleen. In graphs A, B, and C, which are of the spleen samples, the average percentage of live T cells expressing specific genes is higher in the germ-free mice with the old microbiota than in the germ-free mice with the young microbiota.
As living organisms age, T cells within various tissue types receive signals from surrounding cells that cause those T cells to turn into cells with a specific function. T cells have the ability to turn into essentially any type of cell, but once they become specialized, they cannot go back. The spleen data tells us that the microbiota of the old mice contributed to accelerating the rate of aging by accelerating differentiation above the baseline rates of T cell differentiation in germ-free mice.
Inflammation is a characteristic of the body's immune response. Chronic inflammation, however, is the result of the body trying to rid itself of foreign substances or toxins. We see chronic inflammation in the germ-free mice that is triggered by the body responding to a bacterial environment found in the GI tract. This is a notable observation because we know a number of diseases that are found within the older populations of humans that are thought to be caused by lower-level chronic inflammation.
The analyzed tissues in the guts of the germ-free mice were found to have a higher amount of bacterial components in systemic circulation. The evidence for the previous finding where the genes TLR2 and TLR4 were found in an upregulated fashion in the ileum tissue, and this is shown in the graphs pictured here.
The results show that old mice bacteria in the guts of germ-free mice decreased bacterial filtration in the small intestine. This leads to higher amounts of foreign material inside the body, which leads to inflammation. In regards to humans, living a healthy lifestyle can promote gut microbiota that prevents inflammaging. Keeping healthy gut bacteria levels could assist in prevention of illness such as cardiovascular disease, diabetes, and cancer, which increase health costs with their treatment. Well, that's all I have for you guys today. Special thanks to researchers Dr. Hugenholtz and Dr. Van Beek. I'm your host, Daisy, and thanks for tuning in to Daisy's Discovery. Tune in next time where we discuss hot dogs, are they sandwiches?